So in these lectures, we're going to be going through the bioenergetics of living systems. Primarily, we're going to be taking a look at cellular respiration and photosynthesis and how that plays a role in our muscles. So let's first go through what we're going to be talking about. I'm going to go through a quick recap of thermodynamics and what was important. We then want to take a look at an overview of bioenergetics in living systems. So what sorts of reactions are important, as well as the importance of ATP, adenosine triphosphate. Afterwards, we have two main sections of content. One is photosynthesis and the other is cellular respiration. We'll take a look at all of the different processes within each, and we're going to end off with cellular respiration so that we can directly move into some muscle bioenergetics. So let's go through a recap. We talked about thermodynamic potentials last time, as well as the four thermodynamic laws. And at the end, we did present a lot of equations for force flux relations, but most importantly, we want to be able to know the different types of equilibrium, as well as explain the non-equilibrium. So for thermodynamic potentials, this picture's a little bit blurry, but we have thermodynamic potentials being very useful in chemical thermodynamics of different reactions. We want to know how to derive the equations for each potential and describe their meaning. So this is something that you can take a look at previous lectures for, but to get to each of these equations, if I want to get to F, Helmholtz free energy, which defines the amount of energy required for a system, you can have U minus Ts is equal to F. So we're taking away the entropy and only considering the amount of energy required to create the system. Then you can take a look at enthalpy, or very closely related to heat. The internal energy plus PV is equal to enthalpy. And if you want Gibbs free energy, you can start from U, subtract Ts, and add PV to get to G. Or it can be H minus Ts, or it can be F plus PV. All of those work. There are four laws of thermodynamics. The zeroth law, this means that two systems in thermodynamic equilibrium will not exchange any heat with each other. Systems that are in thermodynamic equilibrium are going to have the same temperature. In the first law of thermodynamics, it states that the change in internal energy of a system is equal to the heat added to the system minus the work done by the system, W. So you have this equation, which is very important. Some considerations that we looked at were what happens if you have an isolated system. If you have an isolated system, there really is no heat or work, so delta U is equal to 0. If you have energy being conserved, that means the change in internal energy in your system must be equal to and opposite to the change in energy in your environment. If energy actually enters the system, that means that delta U has to be positive. In order for that to be positive, it means that heat must be added, so Q is greater than 0, and the work must be done on the system. So the work done on the system must be greater than 0, which means this one is then going to be negative. So negative and negative make positive, and delta U is greater than 0. And you can use the same type of mathematics and conceptual thinking to see what happens if energy leaves the system and delta U is a negative value. In the second law of thermodynamics, basically this states that in any spontaneous process, the system is going to tend towards a state of greater entropy or greater disorder. In the third law of thermodynamics, this states that the entropy of a pure crystalline substance at absolute 0 is exactly 0. That's because there is only one possible state for a pure crystalline substance at the lowest possible temperature where the molecules really aren't moving. So if there's if there's a multiplicity of 1, that means a ln of 1 is equal to 0, and you end up with an entropy of 0. Now for the different types of equilibrium. There were 3 that we looked at, plus the non-equilibrium, which I'll present on the next slide. In simple equilibrium, there's basically 2 phases there's a higher concentration of I in A and a lower concentration of I in B. And so that ion I is just going to diffuse across the membrane from a region of higher concentration to a region of lower concentration. In the osmotic flux equilibrium, we now have a bunch of ions or solutes in phase B. So there's a greater concentration of solute in B than A, and the solute is not able to go through the semi-permeable membrane. So it's locked in phase B. In that case, what happens if your solvent S is free to move between your membranes, or between your phases? So in that case, in order to try and reach some sort of equilibrium by reducing the concentration of I, your solvent is going to want to move into region B to reduce that concentration, to try and reach some sort of equilibrium or steady state. Now, for electrochemical equilibrium, what we want to do is take a look at the concentration of specific ions. So electrolytes, like sodium, there might be a higher concentration of sodium over here and a lower concentration of sodium over here. In that case, there are cations and anions on either side, so that usually means that you'll end up with two phases that are electrochemically inert or in equilibrium with each other. So basically, they're all neutral. But what happens if you have a semi-permeable membrane that is highly selective and is unable to allow your sodium and your chlorine atoms to pass through all the time? In that case, even though your phases can be separate, it can be neutral, what happens if you have a larger protein that can't pass through? In that case, you end up with immobile and heavily charged molecules like proteins being stuck inside the cell, which are going to cause a membrane potential to form, because your proteins will never be able to move, but your ions will still continue to pass in and out to try and reach a neutral charge for each phase individually. So let's first start off with bioenergetics in living systems. What is bioenergetics? Bioenergetics describes the energy transformation process in living systems, and the two main ones are going to be photosynthesis and cellular respiration. Living systems are complex and ordered structures. In order to keep their order, they have to exchange matter and energy with their surroundings, and this requires energy. First of all, sunlight is going to get converted to chemical energy that provides mechanical work for muscle contraction, provide active transport in membrane and nerve impulses, synthesis of other biomolecules. So how do we capture this energy? So in terms of capturing energy, there are two types of cells we have to talk about. There are autotrophic cells. It gets energy directly from sunlight through photosynthesis. So photosynthesis essentially produces glucose from water, carbon dioxide, and light. Heterotrophic cells, they use readily made glucose, carbohydrates, fat, and proteins through cellular respiration. And cellular respiration uses oxygen from the atmosphere and produces energy while expelling carbon dioxide back to the atmosphere. Due to these differences, autotroph cells or autotrophs are referred to as producers, whereas heterotrophs are consumers. So in terms of the process, sunlight is going to go into autotrophs to get converted into sugars, and then heterotrophs are going to consume those in order to get that energy. A small subset of autotrophs are actually called chemotrophs. They use chemosynthesis as an alternative method of making food. So basically it transfers chemical energy from inorganic to organic molecules. So here's a nice diagram highlighting the differences between autotrophs and heterotrophs, and what sorts of living organisms belong to each. Most importantly, I want us to focus on two that we didn't mention, bacteria and fungi, which are classified as heterotrophs. Now, maybe it'd be interesting to see if there are any exceptions to these, because sometimes there are some bacteria that can also act as autotrophs depending on their environments. So to power all of these biochemical processes, what is required? ATP is the most important energy molecule, adenosine triphosphate. We really don't have high temperatures, especially for living organisms. We don't have high temperatures or high voltages that we can use for our cells. So our cells also cannot change volume to produce work. So our cells need to produce energy at constant pressure and volume. So what happens is we use ATP to store all the energy, and we use it when we need it. So ATP is a high energy molecule, seen over here, that stores energy required for most biological processes. There are more than 2 times 10 to the 26 molecules, approximately 160 kilograms of ATP that's formed on a daily basis in the human body. That's a crazy amount of weight. Each ATP molecule has an active region. So highlighted over here is an active region. For ATP, there are 3 phosphorus groups, 1, 2, and 3, connected by oxygens. And if you just take a look at these oxygens, they're all negatively charged, and negatively charged groups really shouldn't be that close to each other, which means that we have a lot of energy that can be expelled here. So this stores a lot of energy, and when we convert ATP to ADP, which is adenosine diphosphate, we basically remove one phosphate group. It releases 3.6 kilojoules per mole of energy, which is approximately equal to 1 peanut's worth of energy. Here we explain the energetics of ATP hydrolysis. We can represent this using a change in Gibbs free energy, which we learned previously in Thermodynamics chapter. Gibbs free energy can be thought of as the maximum amount of work obtained from a reaction. So it's very important and useful when it comes to quantifying the energy within chemical reactions. If we have a degree sign over here, it just means that we're looking at the change in Gibbs free energy under standard conditions. Now, to go through ATP hydrolysis, we have ATP and water, H2O, that are added together, and that is going to spark the release of a phosphate group represented by Pi. A glucose molecule within that Pi is going to give us glucose 6-phosphate and another water molecule. So this is another 13.8 kilojoules per mole. Combining these two equations together and their Gibbs free energy, we end up with the ATP hydrolysis reaction equation, which gives us an average of minus 16.8 kilojoules per mole for each reaction that takes place. So now that we kind of know what ATP is and the fact that it can release energy when it's hydrolyzed to ADP, we take a look at another common reaction that takes place in biochemical processes, and that's redox reactions. Note that redox reactions are going to take place between donor and acceptor molecules. Looking at this diagram here, A is going to donate an electron to B. So A is the donor and B is the acceptor. So A donates an electron to B, and because A is the donor, we call it a reducing agent because it gives up an electron and causes the reduction of B. B is the acceptor, making it the oxidizing agent that accepts electrons that causes the oxidation of A. So keep in mind the difference between what an oxidizing agent is and what is being oxidized. Now, looking at a redox potential is given by delta phi. It's used to represent the tendency of a chemical species to acquire electrons from or lose electrons from a redox reaction. It's typically measured in volts or millivolts, and we can use what we've learned before, the Nernst equation, to find the potential. So delta phi is equal to phi f minus phi I for final minus initial states. We have phi equals to phi naught plus RT over F ln of A over D. We also have delta G being equal to this, which we had before, based on the chemical potential equation. Essentially, with a redox potential, we can use the Nernst equation to find the potential, and if the potential difference from beginning to end is positive, then the process is spontaneous because energy is released. If delta phi is negative, then that means the process requires energy. So with some of those talks about ATP and redox reactions out of the way, we can then move on to photosynthesis, which starts with sunlight. In photosynthesis, we can actually break things down into two subsets of processes. One is light-dependent reactions, which require sunlight, and the other is light-independent reactions based on the Kelvin cycle. So what is photosynthesis? Photosynthesis is the process by which green plants and other organisms, specific other organisms, transform light energy into chemical energy. Basically, green plants are going to capture light energy and use it to convert H2O, carbon dioxide, and minerals into oxygen and energy-rich organic compounds for use in nutrients. An overview of photosynthesis. Sunlight is absorbed into the leaf by a green pigment called chlorophyll. Light energy is then converted to chemical energy in the form of starch and sugars. The equation for photosynthesis is as follows. You have 6 molecules of carbon dioxide and 6 molecules of hydrogen, 6 molecules of water. Once you have light or the introduction of photons into the system, it's going to produce 1 sugar molecule and 6 molecules of oxygen. Now, photosynthesis primarily occurs in autotrophs, and within autotrophs, it takes place in plant leaves. Specifically in an organelle called the chloroplast, which has a high concentration of chlorophyll, which helps to absorb light. Each one of these cells may have 1 or 100 chloroplasts, so there's a lot. So within the envelope's membranes, there's a region called the stroma. And this is for a chloroplast. In the stroma, there exists a system of interconnected flattened membrane compartments called thylakoids. So these are called thylakoids, just stacks of thin discs. A stack of these thin discs is called a granum. So that's all 3 of these together would be called a granum, and then several granums connected together are called grana. So these thylakoids or thin discs are similar to the inner envelope membrane of the chloroplast, but most importantly, it is the site of light absorption and ATP synthesis. That's the most crucial component of thylakoids. It contains a lot of proteins that are involved with the electron transport chain, which we'll talk about in a bit. Specifically, there are 2 photosystems, 1 and 2, that are in the thylakoid membranes that govern the light-dependent reactions of photosynthesis. And the question is why? It's because chlorophyll is embedded into these membranes. Now, when we take a look at chlorophyll and light harvesting, why does chlorophyll look green? First of all, it's because it absorbs red and blue light, and they are at 2 different wavelengths. The central part of chlorophyll is a porphyrin ring with a magnesium ion in the center, and there are 2 pigments that will absorb the red and blue light, A and B respectively. And this right here is a bunch of chlorophyll molecules that are embedded in a protein matrix, and we're going to call these photosystems. So we're going to take a look at that after. Now, when chlorophyll absorbs light energy, there are 3 options. Number 1, the energy can be dissipated as heat. Number 2, the energy can be emitted as fluorescence. And number 3, the energy can be transferred to another molecule without radiation, which we call electron transfer. So chlorophyll is only going to trigger a chemical reaction when it is associated with proteins embedded in a membrane, and it requires an antenna system or a photosystem. Basically, this photosystem involves photons hitting your chlorophyll, and those pigments are going to collect light energy and bounce it into the reaction center from non-radiative resonance energy transfer, or RET. Very similar to FRET, but without the fluorescence. The collected light energy, what I want to mention is the collected light energy here is transferred into chemical energy by the reaction center, and this is going to lead to electron transfer once all of the energy is centralized in that reaction center. Now, now that we've kind of explained the passing on of energy to the reaction center, we're interested in knowing the efficiency of photosynthesis. So that depends on the wavelength of the incident light and probability of its absorption. Since photosynthesis produces oxygen, the quantum yield or efficiency is measured by the ratio of the number of O2 molecules produced to the number of photons absorbed. So this can be represented by quantum yield is equal to the number of molecules divided by the number of photons absorbed. So molecules over intensity. When lights of wavelength 650 and 700 nanometers combine, there is an enhancement in the quantum yield, and that we just deem the Emerson effect. So the take-home message is that your quantum yield is going to depend on your wavelength. One thing I wanted to mention here is something related to quantum biology and quantum coherence. Since we know about quantum yield and light absorption now with respect to the reaction center and the antenna system, what happens if there is a plant that doesn't get much sunlight? How can it optimize its yield then? Recent studies in the past decade have actually shown that trapped energy in cells don't just bounce randomly to the reaction center, but rather there's a phenomena called quantum coherence that allows for the cell to predict and pre-test every possible pathway simultaneously before sending the light down a certain path towards the reaction center. So there is quantum coherence in that the chlorophyll and the plant is going to automatically choose the path of least resistance and the quickest path to get to the reaction center. So it's pretty cool that that takes place. Let's now move into the reactions for photosynthesis. There are essentially two kinds, light and dark. We're going to start with light reactions and the two photosystems that are involved. So, the light-dependent reactions use light energy to make two molecules required for the next stage of photosynthesis. There's going to be ATP and NADPH. Light-independent reactions have unique characteristics. Number one, we already mentioned them. They only take place in the thylakoid membrane of the chloroplast because that's where the chlorophyll is. There are two large complexes that have reaction centers, photosystem 1, PS1, and photosystem 2, PS2, and these play a critical role in harvesting light and sparking these light-dependent reactions. Now, something that's interesting is that photosystem 2 actually comes first. And that happens because simply PS1, which happened second, was discovered first. So it's very confusing in terms of the naming system, but just remember PS2, light absorption happens in PS1 first. So there are some, the first thing we want to do is figure out what a photosystem is. A photosystem is basically a system that contains a unique pair of chlorophyll molecules right here. So we call that a special pair that can lose an electron when excited. This electron is then going to get passed on to a primary electron acceptor, and then that gets passed on through an electron transport chain. So there are some key differences between the two photosystems, PS1 and 2. PS1 absorbs best at longer 700 nanometer wavelengths, and PS2 absorbs better at shorter 680 nanometer wavelengths, which is why their photocenters are called P700 and P680, respectively. Now, there are four major protein complexes in the thylakoid membrane. There's photosystem 2, which takes place first. There's a cytochrome B6F complex, photosystem 1, and then an ATP synthase. So it's important to note that there are four protein complexes that are required for the light-dependent reactions. These four proteins are going to work together to ultimately create ATP and NADPH. So first, the four photosystems absorb light energy through pigments. The light energy reactions are going to begin in photosystem 2. when chlorophyll A molecules within the reaction center of PS2 absorb a photon, an electron is released and is excited. So then this excited state of the electron is very unstable, so the electron is transferred from one to another molecule to create a chain of redox reactions through here, called an electron transport chain. So that's where those redox reactions we talked about come in handy. The electron flow goes from PS2 to cytochrome B6F to PS1. In PS1, the electron is going to get the energy from another photon. So another light absorption takes place, or light harvesting event takes place. The final electron acceptor is then NADPH, which becomes NADP. Or sorry, it's NADP, which becomes NADPH. Meanwhile, looking at the far right of the picture, cytochrome B6F and ATP synthase are then going to, so B6F and ATP synthase are going to work together to create ATP. And this process from B6F to ATP synthase is called photophosphorylation. So we're going to summarize this in the next few slides, but the net reaction is that you have 2 water molecules, 2 NADPH, plus 3 ADP and 3 phosphates are going to make 2 oxygens, 2 NADPH, and 3 ATP molecules. So these steps are going to be presented in the next few slides, but I'm just going to stay on this slide so I can explain everything with this picture. So this process that we just explained with the 4 different proteins, we're going to give it a name. We're going to call it non-cyclic photophosphorylation. And that's the usual form of light-dependent reactions where electrons are removed from water and pass through PS2 and PS1 before ending up in NADPH and ATP. This process requires light to be absorbed twice, so that's important to know. In step 1, we have light absorption happening in PS2. Light is absorbed and passed inwards from pigment to pigment. So light is absorbed and passed from pigment to pigment until it reaches the photocenter, the reaction center. And at the reaction center, energy is transferred to P680, exciting an electron. That electron is passed on to an acceptor molecule and replaced with an electron from water, which releases the oxygen that we breathe out. Now in step 2, we start off with this electron. It's going to travel down the electron transport chain, where released energy drives a proton gradient H from the stroma to the thylakoid interior. Your hydrogen or proton ions are going to flow down the proton gradient back up into the stroma and pass through ATP synthase to spur on ATP production. Now, taking a look at step 3, light absorption in PS1. Oh, so there's the ATP. I was trying to look for it. There's the ATP that's created in that second step. In step 3, more light energy is harvested, gets into these chlorophylls, and electrons eventually reaches photosystem 1 and joins P700 with chlorophyll A. Light is absorbed and energy is going to be passed onto the reaction center to excite another electron. And now the excited electron in step 4 is going to travel down another electron transport chain here, and it's going to be passed on to NADP plus alongside another electron to make NADPH. So at the end, you get 1 ATP and 1 NADPH. So these 2 slides just go through the 4 steps. Now, the net reaction is the conversion of light energy into chemical energy in the form of ATP and NADPH. These will then be used to make sugars in the next stage of photosynthesis. So that would be the Calvin cycle and the dark reactions. So we've talked about non-cyclic phosphorylation, photophosphorylation. What about cyclic? It's basically the same thing, but the electrons follow a different circular path and only ATP is produced and no NADPH. So This is a complete picture of photosynthesis in case it helps with the explanations. I won't really go through this too much, just that in photosystem 2, light energy comes in, and then through quantum coherence, you end up with the fastest path towards the reaction center, which excites some chlorophyll electrons, and then it goes through an electron transport chain and eventually produces ATP. That extra electron also continues to the photosystem 1, another reaction center, another light harvesting event, and then through an additional 2 excited electrons, it goes to P700, which then releases electrons that go towards NADP plus the acceptor molecule to create NADPH. Now, once all of that is done, it goes to the Calvin cycle, which we'll talk about next. The Calvin cycle is a light-independent reaction where carbon atoms from carbon dioxide are fixed. Basically, fixation means that the carbon is integrated into organic compounds for use in 3-carbon sugars. It's fueled by ATP and NADPH from light-dependent reactions. So one thing is, because it is light-independent, it takes place in the stroma, so it no longer requires light to take place. Now, looking at this picture here, once again, the steps are presented on the subsequent slides, but I'm just going to stay here so I can explain it through this diagram. So there are 3 main stages for the Calvin cycle. The first stage is carbon fixation. In the presence of carbon dioxide, ribulose 1,5-bisphosphate carboxylase/oxygenase, which we just call Rubisco because we don't want to say that all the time, and 3 molecules of ribulose bisphosphate, RuBP, initiate the reaction. So 1, 2, 3, they initiate the reaction, and for each carbon dioxide that reacts with RuBP, Rubisco catalyzes the formation of 3 PGA molecules, which has 3 carbons and 1 phosphate. So if you have 3 of each, you end up with 6 because each of them make 2. So carbon is now fixed from an inorganic form to an organic molecule. Now in step 2, it's what we call the reduction of 3 PGA. ATP from the previous light-dependent reactions and NADPH are used to convert 3 PGA into 6 molecules of glyceraldehyde 3-phosphate. So that's glucose 3-phosphate, G3P, right here. This is termed a reduction because 3 PGA is going to gain electrons from NADPH. ATP, from ATP, energy is released with the loss of the terminal phosphate atom, so it becomes ADP. With NADPH, energy and a proton are lost, converting it to NADP+. Note that ADP and NADP return to the light-dependent reactions to be reused and re-energized from the Calvin cycle. Now that completes step 2, the reduction step. In step 3, we have regeneration over here. Some G3P molecules go to make glucose, and others are recycled to regenerate the RuBP acceptor. Regeneration requires 3 ATP. So note that in order for 1 G3P to exit the cycle and be used for glucose synthesis, 3 carbon dioxide molecules have to enter the cycle to provide 3 new atoms of fixed carbon. When 3 carbon dioxide molecules enter, 6 of these G3P molecules are going to be made, where 1 exits for glucose synthesis and 5 are used for regeneration. So the next 2 slides are simply just going through the steps for the Calvin cycle. Now let's summarize everything in terms of the energetics of photosynthesis. How much energy is involved in photosynthesis? First, we have the reduction of carbon dioxide, given by this equation, and approximately 470 kilojoules is required. Now with glucose, we then have this equation, And like we presented in thermodynamics chapter, or in the thermodynamics chapter, we have 2,820 kilojoules that are required for this to happen. So what's happening here? For each carbon dioxide, there's a transfer of 4 electrons. The reaction requires 4.84 electron volts of energy per molecule. This energy comes from sunlight, 680 nanometers, and each photon provides 1.82 electron volts. If we require 8 photons to produce one CH2O molecule, we end up with 8 HV equals 14.6 electron volts. So can we calculate the quantum yield? We can, because then we have the total number, or the total energy of energy per molecule, and then we have a total of 14.6 electron volts as the intensity of the light. So then we have 4.84 divided by 14.6. We have a approximate 33% quantum yield. So now let's talk about cellular respiration. We're going to go through a quick overview, just like we did before with photosynthesis, and go through the 4 different stages of cellular respiration. There's glycolysis, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation. Now, with cellular respiration, it's the process by which individual cells are going to break down food molecules like glucose and release energy. It uses the energy released from the molecules of ATP. So with cellular respiration, it really looks like the opposite of photosynthesis, where you have carbon dioxide and water making oxygen and glucose. We take the glucose and the oxygen, and we make carbon dioxide and water and gain energy in the process. So oxygen is required for cellular respiration. It makes it an aerobic process, and it has 4 main stages, which I just mentioned. We're going to go through each one separately. So with glycolysis, it's the breaking down of glucose to extract energy for cell metabolism. You have a 6-carbon glucose that's split evenly into 2 3-carbon pyruvate molecules from the energy provided by 2 ATPs. So you end up with fructose diphosphate, which ends up with G3P, glyceraldehyde 3-phosphate, 2 of them. And the release of energy is used to make 4 ATP. So you have a net gain of 2 ATP. Now, going through each of these, the release of these phosphates, you get these 2 ATP and you also get 2 NADH in total. So just to summarize, even though you've basically removed 2 ATP, but you gain 4 ATP over here, so there's a net gain of 2 ATP. No NADH is used here, but rather 2 are created, so you have a net gain of 2 NADH. With pyruvate oxidation, step 2, each pyruvate from step 1 goes into the mitochondrial matrix. It's the innermost compartment of the mitochondria. You also have this equation here. You have pyruvate being converted into a 2-carbon molecule bound to coenzyme A, known as acetyl-CoA. So you have this over here, right here actually. The carbon dioxide is then released and 1 NADH is generated. And this step actually is usually looped in with step 3, Krebs cycle. So I'm just going to go to the next slide to give you an overview of that. So the pyruvic acid, pyruvate, what's going to happen is 1 NADH is going to be created, but coenzyme A is going to come here and then is going to form a 2-carbon acetyl-CoA complex. And once you start the Krebs cycle, or the citric acid cycle, acetyl-CoA right here is going to combine with a 4-carbon molecule and goes through a cycle of reactions to regenerate the initial 4-carbon molecule to produce 3 NADH, 2 FADH2, 2 carbon dioxide, and ATP. And then you just start back here again, and it goes through in a cycle. In step 4, we have oxidative phosphorylation. This is when NADH, so we have We have the citric acid cycle over here, and it produces an NADH and FADH2. They're going to deposit electrons in the electron transport chain, releasing enough energy to pump protons out of the matrix. So you have all of these H pluses over here. All the protons are pumped out of the matrix. And then you have now a lot of protons that then flow back through ATP synthase over here. So you have an excess of protons. They come out and then they come back in through here. And that is going to provide enough energy for ATP to be formed through ATP synthase. So the question now is, what happens if oxygen is not present? If oxygen is not present, we can still use pyruvate, but metabolize it through fermentation or through lactic acid formation. Fermentation complements glycolysis and makes it possible for ATP to be continually produced anaerobically. What happens that's different is that it oxidizes the NADH produced in glycolysis. The fermentation regenerates NAD+, which can then take part in glycolysis again to produce more ATP. The only problem is that if you do choose to use fermentation, not that you do choose, but your body, if oxygen is not present and uses fermentation, you end up with 2 ATP being created in anaerobic respiration instead of the usual 36 to 38 ATP molecules in aerobic respiration. So there's even though you can go through cellular respiration through fermentation, it's not as effective. Now let's take a look at muscle bioenergetics. We've talked about cellular respiration. Let's now do a quick overview of skeletal muscles, muscle metabolism, and what happens when we're doing hard exercise. So how do muscles contract again? Quick overview. There's going to be an action potential that arrives at your neuromuscular junction. You have acetylcholine neurotransmitters that are going to be released and binds to receptors to open sodium ion channels. So all these little ion channels here. Your action potential is going to travel along to your T-tubules and be created in your sarcolemma. Your calcium is going to be released right here. So all that calcium is going to be released and bind to troponin, which helps initiate muscle contraction. And these thick and thin filaments are going to interact and lead to muscle contraction. All of these processes then follow the sliding filament theory, which takes a closer look at the microscopic level of how muscles contract. But we want to look at the big picture. So muscles will keep contracting as the signals go on. Muscles will stop contracting when the signaling from the motor neuron is going to end. So how is ATP related to muscle contraction? In order for the thin filaments to keep sliding past the thick filaments, the myosin head has to pull the actin at the binding sites, detach, recock, and attach to more binding sites, and repeat the cycle. And that cycle is called the myosin cross-bridge cycle. Each cycle requires energy provided by ATP. So looking back up here at our previous picture, we have ADP and PI. For phosphate, you do need ATP in order to recock the myosin head so that it continuously pushes your filaments so that a contraction can take place. Now, creatine phosphate stores energy in phosphate bonds. In a resting muscle, excess ATP transfers energy to creatine, producing ADP and creatine phosphate. This acts as an energy reserve that can be used to quickly create more ATP. So when the muscle starts to contract and needs energy, creatine phosphate transfers its phosphate back to ADP to form ATP and creatine through creatine kinase. So just to summarize, the whole reaction is going to be catalyzed by creatine kinase and occurs really quickly. So creatine phosphate derived ATP powers the first few seconds of muscle contraction, but it only can provide approximately 15 seconds worth of energy, at which point another energy source has to be used. So that means if you can use it for the first 15 seconds, stored ATP, what happens afterwards? It means that there's going to be some limitations to aerobic respiration. So aerobic respiration cannot be sustained without a steady supply of oxygen to the skeletal muscle. Your muscles are going to compensate by storing small amounts of excess oxygen in myoglobin, presented here. This allows for more efficient muscle contractions and less fatigue. But what happens if you use too much? Muscle fatigue occurs when your muscles can no longer contract in response to signals from your nervous system. So it's really important to know some reasons why muscle fatigue takes place. Number one, your ATP reserves are completely depleted. It means that your muscular function is going to decline. You might have lactic acid buildup, which lowers your intracellular pH. You might also have an electrolyte imbalance, which disrupts the flow of calcium out of the sarcoplasmic reticulum. Now, with anaerobic respiration, if oxygen is not present, pyruvic acid is usually converted to lactic acid through fermentation. So every glucose molecule produces 2 ATP and 2 molecules of pyruvic acid, which can be used in aerobic respiration or converted to lactic acid. If oxygen is not available, the pyruvic acid is converted to the lactic acid, which may contribute to muscle fatigue. And this occurs during strenuous exercise when high amounts of energy are needed, but oxygen cannot be sufficiently delivered to your muscle. So you feel the burn of lactic acid after strenuous exercise. Now, some things to keep in mind. Lactic acid can lower your blood flow and oxygen throughout the body, which makes you tired and lightheaded. Next, it can also cause high levels of lactic acidosis, which can be life-threatening if not taken care of. So just as a summary of what to remember, you should focus on ATP as an energy source, the different redox reactions we talked about, and the differences between a reducing and an oxidizing agent. Photosynthesis, know the main steps and the net reactions. You don't have to know every detail, but remember the big picture. What are the main steps? What kind of components and substances are involved, like the photosystems, chlorophyll, where they take place, whether it's in the thylakoid membrane or in the stroma. Know about cellular respiration, where it takes place, mitochondrial membrane or in the matrix. Once again, don't get into the details too much. We want to focus on biophysics and understand exactly how the processes take place and the different steps that are involved. Finally, we summed everything up through applying cellular respiration to muscle fatigue in case there's not enough oxygen. So know what happens if there isn't enough oxygen, as well as how lactic acid buildup can occur.